Okay, welcome back. We're going to start the second session on specific technologies in the land sector to roll out. Up first is Lee DeHaan from the Land Institute, who's pioneered perennial crops and perennial wheat, and that'll be followed by Jason Hill from the University of Minnesota, who's worked a lot on bioenergy crops and below-ground carbon storage. And then I'm going to fill in for Liz Renato from Evergreening, and we'll follow up with a panel discussion. This panel discussion will have microphone runners, so you don't have to line up, you just put your hand up. And we want to make sure we get some questions from the students up in the back, all right? So think about that. All right, Lee, come on up.
Thanks, Justin, for inviting me here. It's a great opportunity to come and talk about our work, and I think what I'm going to talk about ties in pretty clearly directly with the talks already this morning. What I normally work on is working on developing new perennial crops. So perennial crops would be plants that would live for many years without being replanted, like alfalfa was just mentioned earlier. A plant like that doesn't have to be replanted every year, has a big root system in the ground, and we came into this at the Land Institute for a lot of different reasons. Soil erosion, water, purity, soil health, weed control, all these reasons for developing perennial crops that didn't have anything to do with soil carbon as well before soil carbon, for its own sake of sequestering carbon, was really an issue. So it's kind of now that we're coming back to it, we're realizing the potential benefits. So we're going to try to talk about a little bit what we think is that potential benefit. I'm not a soil scientist or a soil carbon person. I'm a plant breeder. So I'm going to talk more about my work with plant breeding, but we do have a soil scientist on staff, so I picked his brain a bit to put together a little bit of information for you on that first. And I should make use of this. Yeah, so where I come from, Kansas, the prairies are this perennial vegetation that lives for many, many years and covers the ground and built the soil carbon of the prairies. Our richest soils of the earth have the most soil carbon in them, and they come from these perennial herbaceous plants living in these natural communities that produced that through their huge perennial root systems. What we've done in agriculture is on the bottom, till it up every year, usually an annual reset, either by chemicals or by tillage, and that releases huge amounts of CO2. For the most part, once we go to grain agriculture, we lose about 50% of the soil carbon within a few decades. All that goes into the atmosphere. And what we'll be talking about to try to get that back in is going back to a kind of plant community that is more like what created that soil carbon, that healthy soil to begin with. So as I mentioned, there's a lot of reasons to think about having healthy soil biota, having the carbon pumped into that system through those large roots, exuding carbon and then driving that whole soil ecosystem. And it has so many benefits that are well beyond capturing soil carbon. So we have a great potential for synergistic effects to get more than we expect in terms of bang for our buck on transforming agriculture in this way. So this is actually an excavated soil pit where we had the crop that I work with, the first one shown there, intermediate wheatgrass. It's a relative of wheat, shown here on the left side, growing side by side with annual wheat. And you can just see the about 5 to 10 times greater root biomass and how deep it goes into the soil in perennial grasses. This is not a native perennial grass to Kansas. It comes from the same region as wheat, but grows much like the perennial grasses that are native to our region. The reason we're interested in it is because we can harvest a grain off it and use it for human food as a substitute for wheat. So having plants that more closely mimic what the prairie was doing in terms of how much below ground biomass is there is what's going to be the engine that drives that soil ecosystem to do the things important to us. So looking at this actual data from this plant, how much total root biomass can we see in comparison to, say, annual wheat? Usually it's somewhere in the range of up to 5 times as much biomass. Here we have a few different systems that we worked on looking at the change, and we're up to 5 times as great a biomass under that perennial system. What does that mean in terms of capture of carbon dioxide from the atmosphere? We started a study some years back, 2013, using an eddy-covariance tower. You can see these towers mounted here that measure CO2 flowing over that field, and it's well beyond me to explain the math behind that, but we can come up with a net uptake of CO2 out of the atmosphere or release out of the soil over that time period. And actually this field began 3 years before, so this is starting on the 4th year of this field, 4, 5, 6, and 7 of this field. And what we came up with was really high uptake, even though we're several years into it. What we figure is going on is that over time this is going to kind of even out, and net uptake might actually be more like a 1 ton per hectare per year. But what's happening is building of that soil root mass and dead root material and other soil biota building up over a period of time. And so I expect that somewhere in the range of 20 tons per hectare may have been built up. That's kind of a one-time thing. It's not actually going into the soil that quickly, but as we take perennials onto the landscape, this is a really quick hit that we get by sucking up a lot of CO2 quickly as those plants build themselves into that environment. Next question is how much actually is going to the soil? How much soil carbon is being formed? Here we had a 16-year-old field where we looked at an annual rotation compared to intermediate wheatgrass. That's the perennial grass I'm working on, we'll keep talking about. And we had a native restoration, somewhere in the range of 1 ton per hectare per year, which is kind of what we expect based on lots of other data. The native vegetation was not fertilized, and that probably limited the rate of sort of restoration of that system versus a fertilized system where we can have access to greater carbon fixation through more productive plants. Okay, I noticed the numbers on this figure, which we took from a publication here, is we're somewhat different than other ones. I think that people are using different definitions, so don't be confused by that. My definitions are, I think, consistent within this slide. According to these authors, the sort of net increase of the atmosphere is about 3.5 gigatons per year of carbon. And my question is, what could possibly our crop land do in terms of soaking up CO2 in reference to these sort of big numbers that we see here? So we were just looking for the very high end, and so this is my plant breeder's back of the envelope, not to be criticized by real scientists that work in carbon. So crop production globally has released somewhere in 40 to 80 gigatons as we transferred from those perennial grasslands into croplands and sometimes forest. If we were to convert all of the wheat, maize, and soy globally to perennials, using some of the assumptions of how much we think we can take up, we could get about maybe 7 gigatons just in that living plant material, those living roots that would build up over the first 5 years or so. So you could have a kind of a rapid soak up of carbon in several years if you were to deploy perennial plants onto what current cropland very quickly. And then finally, if we grew all the wheat, maize, and soy to perennials, we could maybe take up about 0.5 gigaton per year. And that could go on for several decades as we restore the carbon back to the soil that was lost in that initial conversion. So it's not a panacea, but it's also substantial. So there's actually a significant contribution that could be made if we could take up soil carbon in the way that perennial grasses did prior to conversion to annual croplands. Okay, so this is the kind of native ecosystem that we have in Kansas, And the question is, why can't we just grow this? The trouble is there's not much to eat up here. Cattle like to eat it, but for the most part we don't. So we need our food packaged usually in the seeds. And that can be proteins, carbohydrates, and oils that are concentrated, harvested very easily. And what we need to do is just coax these plants to do that for us. So we did that 10,000 years ago, worked well. We had the wrong plants, unfortunately. We used all annual crops, and they did bad things to the soil quality over time. We need to reverse that and use plants like these, but through plant breeding, get them to put lots of seeds where we can harvest them. So the biggest can you do it story we have is perennial rice. This is a project that's been going on about 30 years now, 25 years. Resulted in crosses between the perennial relative of rice and rice, bringing in the perenniality genes. And we now have perennial rice in China. This is Fengyi who's led that project. Very perennial and now it's quite successful. We have stands lasting more than 6 years. I shouldn't say 6 years, 6 harvests, multiple harvests per year, with yields about the same as annual rice. The properties of the grain about the same as annual rice, and expanding very rapidly, currently 3,000 hectares. Every time we go back to China every year to visit the project, that's increased a bunch, so it's probably a lot more than that now in the last year. So wildly successful so far in terms of the science, and now we're just looking at the transfer of that technology to farmers' fields in China and also other countries. So we kind of see the theoretical possibility. Can we do this with other grain crops as well? And wheat is one that's been worked on a long time, perennial wheat, perennial sorghum, perennial sunflower relatives, and perennial legumes are all projects we have collaborations with. Mine is specifically domesticating a perennial relative of wheat. It's called intermediate wheatgrass. It's on the market as Kernza, K-E-R-N-Z-A, a word we made up because no one wanted to sell intermediate wheatgrass and call it food. So we can grow it on large scale, easy to harvest with conventional equipment, makes the easy transition. It goes into products very well, especially drinkable ones. This is available for sale in this region at Whole Foods out here. Long Root Ale was the first product on the market. Many other beer products have come out. That's kind of because we have a very low supply right now, and it goes nice into a high-value product. You can tell the story on a can. Also usable in many other products, including breads and cookies and things. So how have I done this for the past 20 years almost now? I worked on just domestication. Let's do it the way our ancestors did it, which is lots of cycles of picking the best ones and intermating, replanting. You do that over and over and over again. Eventually you transform the plant. The degree to which we've been able to change the plant to me is very striking. It's amazing. Within about 10, 12 years, I'd found plants that don't drop their seeds. So this is like the basic domestication phenotype. You got to hold on to your seeds so that you can come along and harvest them. They can't all be laying on the ground. Wild type plants, I just took these before I took the picture, knocked them on the table a few times, and we got this pile of seeds versus these that hold on to their seeds. The transformation in things like seed size, quite striking, remarkable. Here's a wild type on the left that has fairly small seeds. We have some plants 2 to 3 times the seed size and becoming sort of grainy and exciting to look at, and that's only in 10 or 20 years of work. But 10 or 20 years is still too slow, so I'm going to talk about how to speed that up. This is looking back over my 12 years of work or so. Breeding cycle, this is grain yield over time in real plots in the fields. This is not sort of theoretical greenhouse yields. These are real field data, more than doubling the grain yield over time. Still very, very low grain yields, mind you, but the rate of increase is really exciting. Same thing for free threshing ability and seed mass. So we can really make changes quickly, but relative to how fast it has to be, we got to pick up the pace. So how are we going to do that? Really evaluating these perennial plants in the field takes multiple years, multiple locations, and if you're doing that, you're only having a generation every 3 or 4 or 5 years. This new technology called genomic selection, very exciting. I don't have the time to explain all the details, but it's essentially 23andMe for plants. As you could have your own genes tested and say, oh, you probably have brown hair and you probably have certain color eyes and you might have this potential disease come up in your life. The same thing with seedling plants. So instead of growing them for years and years, we can develop a model that predicts and says, even though this plant's only two weeks old, we know it's very likely to be one of the highest yielding plants when it grows up. So we cycle very quickly instead and make very rapid gains. Done a couple of cycles now this way, and it looks like we can double or triple our rate of gain using this technique. Other ways of increasing the speed is to say, well, what about wheat? We already have the genes for domestication in wheat, say over here. If we could maybe bring in some of those genes somehow, could we bring those in? And so using sexual crossing, instead of not GMOs necessarily, but we could actually just make sexual crosses between these plants. Could we bring in some important genes? And that's a tricky bit, so you got to do some embryo rescue to make that wide cross happen. Then lots of work as they're very sterile and you have lots of plants with no seed and you try to recover plants. And then you have a whole bunch of weird plants that have a mishmash of chromosomes to sort through. This is something I was trying 20 years ago and there was no way to figure out what we were doing. It's way too expensive. Now with gene sequencing, we can actually do this technique. So for the first time, I'd say this kind of project is doable. We're just doing reduced representation sequencing here. These are the chromosomes of intermediate wheatgrass. There's 42 of them, and these are the two arms, so there's 48 bars here. This is the durum wheat chromosomes. And so we see we've lost some wheat chromosomes and we've retained some wheat chromosomes. And we can see for just a few dollars a plant what they have. So we can do this on thousands of plants now. I'm looking right now for plants that have one chromosome, each of the single chromosomes from wheat. So currently I've found 12 of the 14 possible that have just a single chromosome added. We'll try to figure out what that chromosome does and is there any piece of a chromosome or a whole chromosome that will get us a big benefit quickly. All right, the final thing I want to strategy for kind of acceleration is targeted mutations to make the changes really precisely that we need to jump forward. So I'm going to talk about this as changing a wild plant, but you can also look at this as trying to make perenniality come into a domestic wheat plant or rice plant. If we know what change we need to make, we can maybe make that very quickly and easily. Okay, so for instance, there's a lot of data now on our domestic crops, like wheat. Every 6 months, I think the wheat genome is finished, is published in a high-profile publication, so it's always more finished than last time. Very difficult sequencing the wheat genome. Lots of complex data there, and the idea is if we can understand what's going on in the wheat genome, we could actually make the gene changes in a wild perennial grass and turn it into a domestic grass very quickly. Alongside of this, we've also been sequencing this intermediate wheatgrass. Now it's getting cheaper and faster. We've been able to come up with a pretty good genome sequence in about 5 or 6 years. Okay, so maybe we could just make that change directly in that plant without doing these crosses or the long-term breeding. I'm just going to put up a series of publications because everyone's talking about, well, maybe we can do it now for the first time ever, that we're to the point where we're going to have this sort of third green revolution where we're going to be able to put together the pieces to make whatever plant we can dream of. That's maybe a little bit of an exaggeration. It's going to be pretty tricky. But a number of papers coming out talking about de novo domestication. Maybe we can quickly domesticate plants and very easily do it. Here's an example, and here's another example that was done where they actually did some of the changes in a wild plant and found that they could get a domestication phenotype very, very quickly. These are in model species that are easy to work in. Same thing done with tomato. Very quickly made the sort of 5 gene changes to take something from being very wild to very domestic with just editing those genes. So a couple of years ago, a paper came out led by Michael Palmgren here, who's in Denmark, and we got excited about this because he was saying we can do this with our crop plants because we know the basic genes of domestication in maize and wheat and rice and barley. We can make those changes in wild plants. And so we invited him to a meeting, and he got excited about our work and using this technique in perennial plants. So it's actually a big impact of that. So it's not just let's have more unique plants for the sake of having more unique plants. Let's have plants that can transform the ecosphere and solve a lot of problems at once. So they were able to get a grant to fund this work, and together we wrote a paper outlining how we would do it with intermediate wheatgrass, tackling some of these problems of very small seeds versus wheat. You see here has very big plump seeds, so we know there's a lot of plumpness genes in wheat, and we understand some of the differences between the intermediate wheatgrass form of that gene and the wheat form. And yeah, so here's a list of our candidates, for instance. So these columns showing all the intermediate wheatgrass genes that we have from the intermediate wheatgrass genome sequence that match to known domestication genes. And we go through some ideas about, would this be a really hard change or is this a doable change? The exciting thing was that just in the 3 months of writing the paper, we were having to rewrite sections because what we could edit changed in that time. So I think genome editing is going to allow us to do these things more easily. Okay, so what are challenges to that approach? Basically, we need to have a genome sequence. So we need to sequence a lot of these perennial candidates. We need to understand the basic biology and genetic control of perenniality. That's really poorly understood. So a lot of basic biology research could take us forward a long ways in that. Then if we don't really know all these domestication genes, we could learn a lot more about those. These wild plants do not have genome editing techniques developed. That's going to take a few years on most of them. And then it doesn't do much good to have cool genes in plants if you don't have a breeding program. So there's a lot of other pieces that have to be put together. And so as the plant breeder, I say, well, your biotech's okay, but it still takes a breeder to pull all those genes together. And that's sometimes the more difficult piece. But I'm hopeful that there will be some ways to leap forward and really make a future perennial grain crop that's going to do a lot of these important things for us. Thanks.